So, good morning, dear colleagues. I'd like to welcome you to this morning session on the Keynote lecture. I hope everyone enjoyed the Catholic evening yesterday at the platform in the city. That was really a great fun. And now I'm very happy to introduce our first speaker. It's Professor Kathy Giacomini. She's really an expert in the field what we are hearing about now. She has a PhD and is really a pioneer in the field of pharmacogenomics. She's Dean of the University of California, San Francisco School of Pharmacy. She's a professor in the Department of Bioengineering and Therapeutic Sciences, which she shared for more than 10 years. Over her long career, she has been an active mentor to pharmacy students and graduate students and a champion for women and underrepresented groups in science. Her research on genetic variation in how people metabolize and transport drugs has been highly influential in pharmacology and has been recognized through numerous awards, including the American Association of College of Pharmacy, Volwiler Research Achievement Award, and the American Association of Pharmaceutical Scientists Distinguished Pharmaceutical Scientist Award. She's also one of the few pharmacy-trained elected members of the National Academy of Medicine. Now we will hear about drug transporters, and I think this is really an important field to understand about biodistribution, and I'm really happy that Casey is here today to talk about transporters and drug development from targets through regulation. Casey, please.
Thank you, Ingolf. I guess I want to begin by thanking the organizers for inviting me. I want to thank Ingolf for chairing this, and I want to thank all of you for getting up early and coming to this talk. And I have very much, I don't know how everybody's feeling, but although the weather isn't California weather here, it has been a very fun week here in Glasgow. So that's been wonderful, and the people here are great. So I'm going to talk about transporters in drug development, and I'll talk about a range of research that's going on in my laboratory. And do I point this at this? Do I advance it? There we go. Okay, a signature slide. This slide shows the members of the solute carrier superfamily in the human genome. Each one of those dots represents a transporter. They cluster together. We've clustered them together according to homology relationships. We use 25% sequence homology to cluster them into families. They cluster into 66 distinct families. There are about 495, almost 500 of them in the human genome. Many of these transporters sit on plasma membranes, and they move their substrates into and out of cells across the biological membranes. But there are transporters which are also intracellular transporters, and that's a great area of new research. Mitochondrial transporters, for example, the SLC25, this family, that's mostly mitochondrial transporters. And in each one of the different families, there may be lysosomal transporters or vesicular transporters of sort. The transporters that most people are familiar with, of course, are in the SLC6 family. If you talk about the general scientific community, the SLC6 family has the neurotransmitter transporters, so the dopamine transporter, the serotonin transporter, the norepinephrine transporters. The SLC5 family, though, has become recently more important because it contains a transporter, SGLT2, the target for the gliflozins anti-diabetic drugs. So that's another drug target. In fact, transporters as drug targets is also an emerging area of research as we move from GPCRs to membrane transporters for targeting for human disease. About 120 of these transporters have mutations that lead to Mendelian diseases, and about 6 in the US are screened during newborn screening because they're actionable. The transporters that and about 70 of them are orphan transporters. Orphan meaning, from my definition, that we don't know what they do. We don't know their substrate. We're not even sure if they are a transporter. And I'll be talking about orphan transporters today. We have studied a group of transporters called the major facilitator transporters. They are part of this SLC superfamily. So you can see right there on the left, I have drawn SLC2, which is a part of the SLC superfamily. But the MFS transporters are related to each other in a couple of ways. Most of them have 12 transmembrane spanning domains. They have signature motifs that are common in all of them, so you can pull them out. They have a common transport mechanism, at least those that we have crystal structures for, in that they tend to operate by alternating access to the internal surface and the external surface. So they have a very simplistic transporter mechanism. And the ones that are important for drugs that we've been working on are the 21 and the 22 family. These are transporters that play a role in pharmacokinetics and may be targets for drug-drug interactions. The 22 family is called the organic ion transporting family. And in it are organic cation transporters that play a role in basic drugs, organic anion transporters that play a role in acidic drugs, and then there's some zwitterion transporters that we also study. The 21 family is, of course, known as the SLCO family. That's where the organic anion polypeptides are, and I'll be talking about transporters in both of these families today. Okay, so I decided to divide my talk into 3 sections, which represent the work that's going on in our laboratory, recent and previous work. And I'll start with biomarkers. I'll talk about published work largely, but I will end by telling you where the field is going. We're very actively working in the area of biomarkers. Unfortunately, I'm not going to be talking about our newest work. I knew I'd need this water. And then I'll move from biomarkers and transporters, a very hot and active area of research, to deorphaning transporters. I'll talk about some recent work that we've been doing on SLC22A24, a transporter that we deorphan, and we're now continuing work there. And I'm looking for collaborators there, so I hope some of you will volunteer to collaborate. And then I'll end with deep mutational scanning. We've recently got into deep mutational scanning. I think one of my strengths is that I'm not very good at computation, so we spend a lot of time doing experiments in the laboratory. So let me begin with biomarkers for transporters. So here I am showing you sort of an updated version of the transporters that play a role in pharmacokinetics in the intestine, liver, and the kidney. The ones that I've bolded and made big, for example, in the intestine, we see PGP and BCRP. These are ABC transporters. But they are targets for drug-drug interactions and very much responsible for actually impairing drug absorption, both of these 2 transporters. In the liver, the transporters we're all familiar with, again, shown in bold are OATP1B1 and OATP1B3, influx transporters into the liver, as well as organic cation transporter 1. And over in the kidney, we have organic cation transporter 2 and organic anion transporters 1 and 3, as well as the mates. The International Transporter Consortium, which I was part of founding that consortium in, I think, the mid-2000s, that consortium, just reminding you, was started because there were so many transporters, right? Yeah, I just showed you all these transporters, and a lot of people didn't know which ones were playing a role in pharmacokinetics, which ones were mediating drug-drug interactions. So we thought we'd get a group together, a group which was industry, academia, and FDA, and we got together and we asked questions about which are the most important transporters for mediating drug-drug interactions in particular. So those are the ones I've bolded. The new ones, since we published a paper in 2010 in Nature Reviews Drug Discovery, the new transporters since then that were not in that paper are the mates, mate 1 and mate 2, as well as OCT1 in the liver was not in that paper as a transporter that we should pay attention to during drug development. So the FDA cited our paper, and they developed some guidelines for transporter-mediated drug-drug interactions. And we are now, the International Transporter Consortium has met regularly, and we've updated new information, and we are now put together a big manuscript which we are submitting to NRDD, and well, we'll see if it gets published, but it'll update. It'll have some of the information I'm showing you here. The ones I haven't bolded have other reasons. They're color-coded. We can talk about those in the Q&A. All right, so what did the FDA do with this information now that we learn what transporters are most important, really by mining the literature and looking for clinical evidence? They proposed guidances. They now have an in vitro guidance and an in vivo clinical guidance. The guidances basically direct people in drug development to test their drugs against these transporters, the one I've shown on the previous slide. And if the drug interacts with one of those transporters and meets certain criteria, that may trigger the drug company or the pharmaceutical company to develop, to carry out a clinical drug-drug interaction study. And so, for example, if your drug inhibits a transporter and the inhibitory concentrations are right in the range of the unbound clinical concentrations, do a drug-drug interaction study, because that means that the drug is going to reach the concentrations that are required to inhibit the transporter in vivo. It implies that. But we all know in this room that going from in vitro, no modeling, no simulation, no nothing, just an in vitro study to predicting a clinical study is very dangerous, and you're probably not going to do that well in your prediction. Actually, it does better than you think. I'm showing you data here of 107 clinical drug-drug interaction studies that were actually performed, and they also did the in vitro studies, and the target for all these drug-drug interactions is OATP1B1, this transporter in the liver that brings drugs in. And as you can see, of the 107, 42 or 35, 70%, let's say, 70% of them actually were true positives and true negatives. That is, they either predicted the drug-drug interaction was going to occur and it did, or they predicted it wouldn't occur and it didn't. But there's a number of false positives and false negatives, about 30%. So we could do better. And at the point when these data this was 27, we're getting published, and people were realizing we had a long spread between those in vitro studies and the clinical studies, we began to think about biomarkers. We and many others began to think about biomarkers. Can you find an endogenous substrate of a transporter that's just like the drug, and it may tell you something about whether a drug-drug interaction may occur or other things about the transporter activity? And so let's go to OATP1B1. As you can see, it transports a number of drugs, including statins, anti-diabetic drugs, anti-cancer drugs, and antiviral drugs, and many drugs. And it's an uptake transporter in the liver. It has a partner, OATP1B3, but I'll talk primarily about OATP1B1, which is the more highly expressed of the two transporters in the liver. So this is beautiful work by Mikko Niemi. I don't know if he's here. I know he's here at the meeting. I don't know if he's here in this room today. This is beautiful work done by him, where back in the early 2000s, people discovered that OATP1B1 had a polymorphism, a non-synonymous polymorphism, valine to alanine at position 174, which had reduced function. This was work done, I think, in Richard Kim's group. And then people did clinical studies, and as you can see, people who are homozygous for the alanine have higher drug levels of simvastatin. Why? They're not getting the simvastatin into their liver as well as people who carry the reference allele. Therefore, the drug levels are going up. So this is the kind of data that make you think, well, if there's an endogenous substrate, people who have these genetic variants, who carry these alanine alleles, also will have higher levels of that endogenous substrate, whatever it is. So let's discover the endogenous substrates of OATP1B1 and the other transporters. And so we mined, at that point in time, we mined publicly available genome-wide association studies, where they had, at that point, I think there were like 7,000 DNA samples that they took from 7,000 individuals, and they genotyped them on a big, genome-wide platform. And they also did metabolomic studies. So they measured a whole bunch of metabolites, maybe 400 metabolites. And we asked a very simple question: which metabolites associate with OATP1B1 valine? And whatever those metabolites are, they might be substrates of the transporters. So these are data that we obtained back then. We found 12 metabolites associated at genome-wide levels of significance with OATP1 valine to alanine, and you can see that here. And you can see these p-values are absolutely spectacular. They're everybody's dream if you're in pharmacogenomics, 10 to the minus 300, and in these Manhattan plots. And the highest peak point was the valine to alanine, that particular variant. So that was also proof. We did a whole lot of in vitro studies showing that each one of these may be substrates or actually substrates in vitro of OATP1B1. They could associate and not be a substrate, and I'll be showing you data later on when we get into our deorphaning, where we have a lot of pretty high associations of non-substrates because of other reasons that we'll talk about. So these were great work. We then started collaborating with Deanna Kretz, my next-door neighbor at the time, and she had carried out a clinical drug-drug interaction study, OATP1B1 mediated, with pravastatin and cyclosporin. It was in 20 people. They administered pravastatin alone, and then pravastatin plus cyclosporin. That's the data on your left. And you can see that cyclosporin administration in these 20 people crossed over causes pravastatin levels to go up, presumably through inhibiting OATP1B1 as one of the major mechanisms. We then measured the biomarker with the idea that, well, a biomarker, and we took HDA, which is, by the way, a specific OATP1B1 biomarker. It is not transported. We couldn't find it transported by 1B3. So it's a specific OATP1B1 biomarker, so we decided we'll take a look at that. And we measured that biomarker in her samples, which were sitting in the freezer next door. And you can see that HDA levels go up just a little right here, and we think that's because pravastatin itself is inhibited, because pravastatin is still on board. So HDA levels are going up here. We think pravastatin is inhibiting that, and then they come back down. But in the presence of cyclosporin, the levels go way up. So it's a no-brainer to think that, well, if you have a new drug and you're doing a phase 1 clinical trial with your new drug, and your new drug happens to inhibit OATP1B1, HDA levels are going to go up. All the endogenous substrates of OATP1B1 that are true biomarkers, their levels will go up. And it's also a no-brainer to understand that not only will their levels go up, but possibly with in vitro kinetics and your clinical data, and how much that endogenous biomarker is going up. And if you know something about the kinetics of the endogenous biomarker, you may be able to predict magnitudes of drug-drug interaction. Not will you have a drug-drug interaction, but what would the magnitude be, especially if you put it into PBPK modeling. And that's a hot area of research right now in the biomarker studies. So where are we in biomarkers? Well, we're far along in the biomarkers, but there are some major gaps, which I'm going to highlight right now. So first of all, several of those transporters that I showed you, the ABC transporters, PGP and BCRP, do not have validated biomarkers yet. So we need discovery work in the area of transporter biomarkers, that's for sure. Secondly, these biomarkers have been applied quite a lot to drug-drug interactions, a little bit to pharmacokinetics, but you could learn a lot from these biomarkers about a lot of effects of, for example, disease. If I want to know, what's the effect of kidney disease on OATP1B1, I might be able to measure the biomarkers, provided they're not cleared in the kidney or something like that. There's work to be, you have to do a lot of thinking there, but you could use the biomarkers to look at effects of disease, aging, developmental changes, and things like that. We now have a way of measuring transporter activity and expression with these biomarkers. And the next area of research, so we need to discover biomarkers, we need to apply biomarkers to understanding disease mechanism and things that we in clinical pharmacology are interested in, special populations. And we need to do more modeling and simulation to begin to learn how to quantitatively predict drug-drug interactions. So this is a very hot area of research in the pharmaceutical industry as well as in academia. I'm going to conclude it and go on to my next area. So I'm going to segue now to deorphaning transporters, and we've been focused on deorphaning transporters in the 22 family. And we're actively working in this area. And again, there's some work that I won't be presenting today, but I'll present some published work as well as some new work that's not yet published on deorphaning one transporter in the family, 22A24. We've de-orphaned, we've got 2 published papers out. One where we de-orphaned 22A15, a zwitterion transporter, which loves, by the way, ergothioneine, your favorite antioxidant, for those of you who like to take things that you think will help you live longer. That's eat mushrooms. Anyway, so there's that transporter, and then 22A24, which we're actively involved in doing research. I'll tell you where we're at there, and I do need collaborators who are interested in sex steroids that might help us unravel some more about this transporter. Okay, so how do we de-orphan transporters? We apply a number of different methods, and we use, of course, publicly available data sets. So metabolomic GWAS, the kind that I just showed you to discover natural substrates of OATP1B1. You can use that to ask the question of what's a natural substrate of an orphan transporter if the orphan transporter has a polymorphism, okay? There are many that don't, so that's a stumbling block. Phylogenetic analysis, that'll help you a lot. I'll show you how we did that. We do use some structure information, and if in the Q&A, if there's time and somebody asks, I'll tell you how we've used structure information to give us clues on how these orphan transporters are functioning. We also, our gold standard is we use isotopic uptake or metabolomic analysis. You can take cells, express a transporter, expose it to serum, and see what it takes up. Measure the inside, the cell lysate, and see what you get. Confocal microscopy is an absolute must. We GFP tag all the transporters in the 22 family, including all the orphan transporters. We express them all in cells, and we ask the question, which of them are on the plasma membrane? Why? Because I told my people in my lab, I don't want to de-orphan a lysosomal transporter. We don't even know how to study them in my lab. Let's look for the ones that are on the plasma membrane and see if we can de-orphan those. So confocal microscopy gives us an idea of some of those transporters that are orphans are not on the plasma membrane. They're not going to be easy to de-orphan. And then we do deep mutational scanning after we know what the transporter does. So here's the phylogenetic analysis of the SLC22 family in the human genome. There are some 30 transporters in this family. It's a pretty big family in the 22 family. They tend to cluster together according to their substrate specificity. So the ones shown in green at the top there are the zwitterion transporters. And those of you who know this family know that octan2, the carnitine transporter, SLC22A5 is one of those. 22A4 is a known ergothioneine transporter, zwitterion also. And we de-orphan 22A15, another zwitterion transporter that really loves ergothioneine. And then in blue are the organic cation transporters, organic cation transporter 1, 2, 3. Black means we don't know what they do. Those are the orphans. We're not sure what they're doing, so we colored them black. And red are the transporters that take up organic anions, and you can see that 22A24 clustered there with the red group, with the organic anion transporter. So our first hypothesis is, okay, it's an organic anion transporter of some sort. What is its specific substrate? We needed to learn that, but at least it clustered there. I think nothing else there. One of the reasons that we decided to start with 22A24 is that it has a common stop codon. Well, you can imagine that if some transporter has the common stop codon, that stop codon, that transporter with the allele with the stop codon is likely not to function, and therefore may associate with some metabolites if, in fact, the transporter is playing a role in their elimination or their absorption. This stop codon is at position 501. It normally was a tyrosine. It's mutated and becomes a stop codon. As you can see in the global plot down there that we drew, the stop codon is the major allele. In most populations. It's like 75% in Europeans and many other populations. The stop codon is 75%, with the tyrosine allele being the minor allele. So we are losing this transporter. I'm not showing you the data, but we showed that, in fact, the stop codon does not function. So we are losing this transporter, at least in the function that we've de-orphaned. But some people carry functional allele. Actually, I'm giving you the RS number for those of you who have 23andMe data and like to look up whether you have the stop codon or you have the major allele. You can look it up. In my laboratory, everyone that had 23andMe data were homozygous for the stop codon, except for me. I have one tyrosine allele. God knows it probably is something that makes me as crazy as I am. So, okay. So, all right, so that was a good thing because we could then associate the stop codon. So we asked, what metabolites does that stop codon, does that RS1123 associate with? And we mined these data, these 7,000, the data sets, as you know, have grown. But we mined it and just simply asked what's associating. It's just really a lookup for us. And very nicely, we found steroid glucuronides in particular. Probably mispronounce it, but I say ethiocolanolone glucuronide and androsterone glucuronide at very good p-values, 10 to the minus 55 and 10 to the minus 38. Also, parent steroids were associated at genome-wide levels of significance, progesterone and testosterone. Not at the time testosterone. We didn't publish testosterone. That wasn't associated, but I just included it here to let you get the feeling that you're getting an association with both the glucuronide conjugates and the parent compounds with the stop codon. So our hypothesis, again, another no-brainer at this point, was this transporter is a steroid conjugate transporter, likely may also transport steroids, and particularly these sex steroids. So we did some studies, and now we transfected this transporter, as I said, in cells, and we're looking at uptake. I think this was with LCMS-MS studies, although it might have been isotopic uptake, can't remember. And you can see for androsterone glucuronide, we have a 270-fold uptake over empty vector. So it is indeed a substrate of this transporter. And ethiocolanolone glucuronide, a 30-fold uptake. So that did say, we tried progesterone. We didn't know testosterone at the time, and anyway, it's hard to get testosterone in the United States. At least I can't get it very well without filling out a million different papers because it's considered a drug of abuse, and nobody in my lab will fill out the paperwork, so we've yet to test testosterone. I need a collaborator here. So, and we did kinetic studies also. And since then, other associations have appeared with the stop codon and a bunch of different steroid glucuronides again, and if this is when testosterone appeared and appeared in some other studies as well. So there's more to be done. We now wanted to know, besides these glucuronide conjugates of sex steroids, what else is this transporter taking up? So what about sulfates, sulfate conjugates? So you can see it takes up ester sulfate and DHEAS. That's the uptake in the cells expressing 22A24 versus empty vector. It takes up, of course, more glucuronide conjugates, and it takes up bile acids. Bile acids, as you know, have a steroid backbone, and they have a carboxylic acid, so it has an anionic component with a steroid, so they're substrates. We have a feeling the transporter is not playing a role in vivo in bile acids because we could never find an association with a bile acid in that stop codon, but the transporter does take it up. So those are sort of the range of substrates that we looked at. We always get into, because, you know, anyway, we get into evolution. And so we thought we'd look at why we're losing this transporter, okay? Why do we have this stop codon? What's going on? So in the Neanderthal and the Denisovan, they both, those hominids, both have the transporter and they have the stop codon. There's some with the 501 tyrosine and some with the stop codon. So they, like us, have some kind of, it's a polymorphism for them. In old and new world monkeys, the transporter's not even there. So we couldn't find it, so I said, we're doing something wrong, we can't find it. So we started collaborating with some evolutionary biologists, they can't find it either. The gene is gone. So in these old and new world monkeys, you don't even see the gene. Where it is, I don't know. Mouse lemur, it's there. And you can see it's also in donkey, rat, and rabbit. And it's in higher order great apes. It's in the great apes. We saw the transporter there, and it looked, and the stop codon was not in the chimp. So we thought, okay, the great apes have it. We constructed some of these and expressed them in cells to see what they did. And it looks like they also like steroid conjugates, and you can see some species difference. Like if you look here on the right with estrogen sulfate, there's the mouse lemur really likes estrogen sulfate. Some of the other species like it okay. So there are species difference in the function of the transporter, which is kind of interesting. One of the works that we're currently doing in my laboratory, we couldn't de-orphan a particular transporter, which I'm not going to talk about today, but hopefully soon. We had to go to a species version to really de-orphan it and then back to the human. So that's an interesting story. All right, so what is the transporter doing and why is it associating with different levels of these steroid conjugates? So we put it's probably likely in the kidney or the liver. That's what those are organs of elimination. It's probably involved in the elimination. That's what we thought. We first, as I said, GFP tagged it. It's on the plasma membrane. There's GFP alone. Looking for transcriptomic data for a transporter that has a stop codon is not easy. Why? Nonsense-mediated decay. The transcripts are gone. So you hardly find the transporter anywhere. So, you go to GTEx. You can't find this transporter there. But we were very lucky. There's always a lot of, I didn't put that as one of those methods that we're using, but one of the big methods is called dumb luck. So we lucked out here, and we found the transporter in single-cell RNA sequencing data, and it's in the proximal tubule in segment 3. And that's where we see it. We think it serves in reabsorption of steroid conjugates back into the blood when you don't have the stop codon, when you have the 501 tyrosine. And the reason we think that is that people with the stop codon have lower levels of testosterone, progesterone, and the steroid conjugates. So we think what happens, they have the stop codon, they're spilling the steroid conjugates in the urine. And people who have the functional alleles have higher levels of these steroids and steroid conjugates. So that's what we thought. Since then, we've done some work. Oh, yes. The stop codon is also associated with diseases that you might associate with steroid dysregulation, like acne and certain cardiac defects. So we've now expressed the transporter in polarized cell lines. This is pig kidney cell line. And we've grown it in transwells. And we've put a substrate, this is taurocolic acid, in the apical compartment. So we've grown it, we've grown it right here. And we put taurocolic acid up here. And you can see we get a very nice uptake from the apical compartment. That is inhibited, and the uptake is over empty vector, shown in the open bars. It's inhibited by an inhibitor, so that looks like it's on the apical side. We put it in the basolateral compartment. We don't see anything. So our hypothesis is what we originally hypothesized, that the transporter is, we're secreting these steroid glucuronides. OAT1 and OAT3 love steroid glucuronides, so they're likely secreting them into the urine. But this transporter is downstream in the lower proximal tubule and is functioning in some reabsorption, some recovery, like we recover glucose and something like that. And then you get the steroid conjugate. There are glucuronidases in the kidney, and you regenerate the parent steroid, and that results in some phenotypes that we think we might see. So that's where we are with this transporter. Let's go now to the chimp. So I got interested in the chimp, okay, because the chimp had the transporter. It looked like it. But the chimp had 551 amino acid transporters, and we have 552 amino acid transporters. So that was interesting. And the chimp is losing or lost a leucine at position 237. So we put it into cells. And when you did, you can see from the confocal microscopy study here at low magnification that, the 551 isn't on the plasma membrane. It's kind of just in the cell. And we couldn't get it to function. So we added back the leucine at position 237 just for fun. And when we did, as you can see, it took up substrates, just like the human one. So there's the chimp and here's the human. Not as good, but it's taking it up pretty well. So somehow when we first saw these data, we thought, oh, the chimp has lost the gene entirely. They all have this leucine deletion at position 237. But then we started collaborating with some evolutionary biologists and primatologists in France. And those guys, Thomas Marquet-Bonnet, who I have yet to meet in person, but we've interacted a lot over Zoom, have access to primate colonies in Africa. And what they found was very interesting. So let's look at 2 different colonies. Let's look at the ones over in West Uganda and Zambia, here and here. There are 19 of them in the colony that have been sequenced. Those 19 all have the 552. They have a leucine at that position 237. But then let's look at this colonies over here in the Ivory West Coast of Africa, and 11 of them are homozygous for the deletion, which is what we thought. So it looks like the chimp has also got a polymorphic SLC22A24, and they're losing it also. They're losing it also, though some colonies retain it and some don't. So we found that rather interesting. So I should conclude and ask you and tell you where we're at. So what we're doing now in my lab is we've made a knock-in mouse. Mice don't have 22A24, at least we don't find it. So we've knocked in the human one into the mice under a kidney-specific promoter, and we're doing some work there to characterize what it does. Is it really involved in steroid disposition of some sort? We think steroid reabsorption. We're also collecting data from humans. So one of the things we find is metabolomic data, but it's hard to find renal clearance data, where you have both metabolites in the urine and the plasma. So we're looking for, again, if there's a collaborator here who happens to have that data, we're looking for that to see if, in fact, it is a renal clearance mechanism for the association. And then we're doing some other studies. So that's where we're at. We're also deorphaning. We've got another one almost totally deorphaned. So hopefully in a few months we'll be able to publish that paper, and I'll be able to talk more about it. I now want to go to the last part of my talk, deep mutational scanning, and we're going to focus on one of my favorite transporters, organic cation transporter 1. This is a transporter in the liver, the most abundantly expressed transporter in the SLC superfamily in the liver. And it sits on the sinusoidal membrane, and it is very promiscuous, so it interacts with a number of drugs. We personally think its major endogenous substrate is vitamin B1 thiamine, and we have a lot of papers showing thiamine interactions with this particular transporter. It's highly polymorphic in terms of reduced function alleles. Not a stop codon, but a whole number of reduced function alleles. And those reduced function alleles, beautiful work from Mladen Cvetkov and his group in Germany showing these polymorphisms associate with increased drug levels. And you can see this is secondary structure of OCT1, and these are some functional studies, each one carrying an OCT1 allele, a non-synonymous allele. And you can see there's some reduced function ones and some normal function ones as well. This is with MPP+. We've done this study with others, and others have done studies with many other substrates. I'm going to skip this slide. Just know that the polymorphisms that have reduced function associate with higher drug levels. So I'm going to take you to our deep mutational scanning work. But before I do, I want to first just take you to Nomad, the database, which has lots of DNA samples. And we decided to mine it for what non-synonymous or missense variants are there in OCT1 in that database. And as you know, as we've heard, and it's true, everything's skewed towards European. So there are a lot of European samples in that database. I think there are 56,000 in this version, samples, DNA samples from Europeans. And those are shown in the brown there. And they have, and there's a total of 270 missense variants of OCT1 in Nomad in this version, total. And you can see they're in each of the ethnic groups. The reason they may be present at lower numbers in some of the ethnic groups, for example, East Asian, is that they don't have very many samples of East Asians, not that they have fewer variants, although we're going to learn something here about that. So each ethnic group has shared variants, which they share with other ethnic groups, and they have their own ethnic-specific alleles, each ethnic group. So that was made in Ann Daly's, that point was made in Ann Daly's session the other night, and again by Munir yesterday, that, we need to look at different ethnic groups more and more to just see, because here are these different variants that they have that no one else has. If you don't look at them, we'll never know anything about these variants. So this just is setting the stage for our deep mutational scanning. So we started to collaborate with a guy at UCSF, Willow Coyote Maestas, and he had worked with Doug Fowler. So he had good expertise, and he was working on GPCR deep mutational scanning. But we said, hey, would you like to, collaborate with us, and we'll collaborate with you and learn about OCT1. So the method is basically you take the cDNA for OCT1 and you mutate every amino acid to every other amino acid. But each OCT1 transcript has only one mutation. So we generated 11,634 mutated OCT1s, each with one mutation, and each amino acid being mutated to every other amino acid. We don't mutate the methionine at the start codon, so it's 554 rather than 555. All right, so now we have this gamish, and we transfect them into cells, okay? Now we have a plate full of cells, and we want to figure out which of the transporters are functioning well and which ones are not functioning well. So we have a platinum analog that we had in my laboratory because we were studying platinums a number of years ago that loves OCT1. And it will kill cells that express OCT1, but if the cell doesn't express OCT1, they're resistant to it. So we exposed this, first we sequenced. First we made a plate, and then we sequenced, so we knew what was there, which of these were abundantly expressed in these cells that had the OCT1, and which ones were not. And then we exposed the cells to this platinum analog. It killed off, or started to kill off, cells that have a functional OCT1, because the platinum can get in and then kill the cell. Things that have a non-functional or reduced function allele of OCT1 are resistant. So at the end of the experiment, you have a whole bunch of cells likely with reduced function transporters and a few with OCT1s that have some function. And then we sequence at the end and we compare. Now, so we now have data, and this is in bioRxives for anybody who's interested. It's up in bioRxives. We've started in the highest journals, and we're going down to get our paper published. But right now, it's in bioRxives for anybody who's interested in reading the results. But I'm going to show you what I find interesting in terms of these natural variants, those 270 variants, because we now could just pick them out and see how they were functioning. Whoops, I'm getting confused here. Okay. So here is, we're able to score their function based upon how frequent they are, that they are in the gamish at the end after exposure to OCT1, after exposure to the platinum. So what we've done here is, this is 80% activity, this is 20% activity. All these mutant transporters function what we call sort of normally between 80 and 20%. There's a whole bunch of reduced function alleles of the 271. So of the 271 natural variants that are found in NOMAD that at least one person on Earth is carrying, there's a bunch of reduced function alleles, and there are even a few gain of function alleles, a little bit gain of function. We see that when I look at the 11,000. Same pattern. There's a few gain of function, a lot of reduced function. Not a lot of gain of function, but a few. And then a whole bunch in the middle. When we go back and look at the ethnic groups, what we find is each ethnic group has their own complement of reduced functional alleles. So this is 20% here. So all of these have reduced activity, 20% or less. And so each ethnic group has a complement of reduced activity alleles, not surprisingly, but they do. We then did something interesting, which was we said, okay, well, what is the frequency of reduced function alleles if I add them up? So if each one of these has a frequency, like this might be a singleton, so it's 1 in 10,000, but this might be present at 20%. So we added up the frequencies in each of the ethnic groups, and what we find is that many of the ethnic groups have pretty high frequency of reduced functional alleles. I mean, this is a gene where 30% allele frequency of reduced function alleles in Europeans. That's pretty high. Most genes don't have 30% reduced function alleles. Latinos also. And East Asians have less than 1%. So it is very dependent on ethnic group. So where are we going with this? Well, right now we're writing this paper. This kind of data we feel is grist for the mill of those people, and we heard a nice talk the other night, who are doing deep learning methods in how these variant transporters may function. So when we go to a day where there will be sequencing and we will uncover these variants, we can predict their function. So we think these data can be used by those people who are doing computational models. We also believe in transfer learning, where they can take the data we've generated for OCT1 and maybe apply it to OCT2. There'll be a lot of commonalities there and get a pretty robust prediction there. So conclusions. PheWAS with metabolomic data can be used to discover biomarkers for transporters. A number of endogenous substrates of OATP1B1 we're discovering using PheWAS and validated in vitro and in clinical studies. 22A24 is an organic anion transporter that prefers steroid conjugates. A common stop codon associates with lower levels of sex steroids, testosterone and progesterone, through the mechanisms that we showed you. Chimp 22A24 has a common polymorphism that also reduces function, and deep mutational scanning is being deployed to functionally annotate all mutations in 22A24. East Asians have a low allele frequency of reduced function alleles, and others have very high allele frequencies. And so what are the conclusions? Well, with biomarkers, let's represent a promising and mechanistic way to understand the effect of various clinical conditions and perturbations on transporter function. If we get specific transporter biomarkers, not for 3 transporters, but for 1 transporter, we can really begin to understand mechanism. Deorphaning transporters should be a major goal of transporter biologists and pharmacologists with, 25% of the transporters, not 20% of the transporters. When we don't know its function, it's hard to learn a lot about biology and pharmacology. And then DMS, deep mutational scanning, can be performed for each pharmacogene that is being assayed in pharmacogenomic implementation studies, and keeps using that. And these are the people involved in the study. My long-term colleague, Sukhwa Yi, my good friend, Deanna Kretz, Thomas Marquet-Bonnet, who I love to meet and go to France, and I'll actually tour the primate colonies in Africa, and Willow for bringing deep mutational scanning to our access, and his postdoc, Christian, and funding from NIH and some from FDA. Thank you, and I'm happy to take questions.
Thank you very much, Casey. This was really exceptional and a big overview, but also detailed data. And I like the term deorphaning transporters because that's really science. So thank you very much indeed. The paper is open for discussion. There's a question. Please identify yourself.
Hi, my name is Manan. I'm final year PhD student. I also work with transporters. I have a very basic question here that as you mentioned that, okay, there are too many orphan transporters that we have not deorphaned. According to you, what are the major challenges in this process of deorphaning transporters?
Well, many of them, so there are many. So many of them won't be on the plasma membrane, and they're going to be challenging to deorphan. Lysosomal transporters, mitochondrial transporters. Another group of them, they might be orphan transporters because the genes are gone. We don't really have them. We don't know that we don't have them. We see the gene there, but maybe they don't have function. So we're trying to throw everything at them, and there's no function. So that's another problem. I guess a third problem is, we don't have access to substrates that are, when you do metabolomic, you only get this complement of substrates. But if I ask them, well, can I see what thiamine levels or norepinephrine levels or, some smaller, they're not measured, or maybe they are, but they're going to cost me a lot of money to figure out. So that's another area of challenges. We don't have access to the substrate.
Okay.
So thank you.
So please.
Yeah, great talk. My name is Bhagwat Prasad from Washington State University. So this regarding SLC22A24, you mentioned it's expressed in the kidney. Is it expressed in other tissues of, for example, relevant to steroid disposition, prostate or testes or other tissues where this could be present and could be functional?
Yeah, so that, I can't even tell you how much, that's been a headache in my laboratory. Why? Because, as I said, nonsense-mediated decay has caused a problem when you do the transcriptomics. So we've gone to proteomic data, okay? Can we find it expressed proteomically? So we're working with Per Arders and others who do, we should start working together. But anyway, so we're trying to find the transporter. So we don't see it all the time. In the liver, for example, that's where we think it could be, so we don't always see it. So the answer is going to be more detailed proteomic studies to find if the transporter is there or not. Right now, we think it's in the kidney, and we're not seeing it in other tissues. Probably it's in the liver. But.
Great. So when you said we are losing this gene, are there similar, so we work on UG2B17, and we were talking yesterday about this thing. Could be that trans kind of linkage disequilibrium where we see multiple transporter and proteins or other enzymes we are losing simultaneously. So any thoughts on?
It's funny, that is a way you could lose genes, but I feel like Bhagwat and I were talking yesterday, he studies UGT 2B17. And that also likes steroid glucuron. That glucuronidate steroids, and the same steroids, and we're losing that one too. So I somehow feel there's a biological mechanism. It's not that they're connected on the same chromosome or they're in linkage. There's a biological mechanism for either we don't need it anymore, I don't know. So I feel like we have to get together and look at coevolution. Of course, neither of us have the expertise to do it, but we should try to look at coevolution of these things, because I have a feeling it's like that.
Thank you.
Thanks.
Roland.
My name is Roland Seifert from the Hannover Medical School in Germany. It was a very entertaining presentation. And as a pharmacologist, I asked the question, do you have specific inhibitors of some of these transporters that we could use in functional studies?
Yeah, so one of my things with biomarkers is that you can find specific substrates for transporters. You can, but specific inhibitors are tough.
Yeah,
and I spent, I burned out 2 graduate students on looking for specific inhibitors in the kidney. So we found specific inhibitor of MATE1 over MATE2. We did the clinical study, and we couldn't see a drug interaction. Why? Because maybe you do need to inhibit them all, You're not going to see too much of one. So specific inhibitors are tough, but we need them. We need them.
Do you have a strategy?
No, not for that. I just start whining. Thank you.
Please.
Hi, I'm Ken Jacobson. I'm a medicinal chemist at NIH. Maybe I can help you with some inhibitors.
Oh, that'd be good. Okay.
But I'm wondering, are there transporters on the nuclear membrane? You didn't mention anything about that.
Could somebody else in this audience know of nuclear transporters? I know there's a big, I got guys at UCSF who are studying the nuclear pore, and there are all those pores and stuff like that because big molecules get out. So I'm not sure. I think there are. I actually think I have seen that, but I'd have to go back and think about it. Thank you.
I have a last question, because we learned that some transporters have stop codons, and we are able to live with or without them.
Yes.
The question is, when you look for the sum of uptake transporters, is it possible to look for all of them, or a number of them, to look which are we able to survive and not, which are really essential?
Yes. So I think that's very important, and knockout mice have given us some clues where, you knock them out, and gosh, a whole lot of transporters that don't have stop codons, you knock them out and they're fine. However, as you and I might know, that if you put them in certain conditions, so asking that question is fine, but could they survive in starvation? Like OCT1 is a perfect example. You knock OCT1 out, and those mice do better in starvation than without. Gave those mice a selective advantage. So I think it has to do with what's essential, but what's essential in a particular condition. And many of these xenobiotic transporters, they take up a lot of things. They're very promiscuous. They're redundant. We can do without them. So anyway, okay.
Okay. So thanks a lot again. Please join me in applauding her again.
Thank
That was
you.
an excellent talk.
Thank you.
So I close this session and wish you an exciting congress today.